Good morning, everyone. And now let me try to pull out my screen. Okay, I think my screen is up. So hopefully my speakers and also my microphones and also my slides here all looks okay.
Yeah.
Yeah, so if you have any concerns or any questions, feel free to drop it into the chat box. Yeah, we will go through, if let's say there's any question, we will go through the questions at the end of the session. All right, so for today, it's going to be a kind of simple sessions. So assuming we are working on metagenomics related projects, so what we should be expecting or what kind of data that we can get from NGS based metagenomics projects. So for these sessions, we will be going through some metagenomic solutions, especially the one that provided by Novogene. So Novogene is our partner, and then they do provide a lot of different sequencing solutions, especially for the metagenomics projects. Actually, I have two screens, so I might be switching here and there. So continue on. So I would also want to show you how a project looks like if, let's say, we are running some metagenomics NGS-based projects, and then what we should be expecting from a metagenomics sequencing project. And then for each of the projects, we'll be getting reports to kind of have an overview of how the projects goes. So this session actually we will be more focusing on looking into the bioinformatics analysis project reports. So to see what kind of data or information that we can get from a metagenomics projects. We will be more focusing on the Amplicon or like the 60S or ITS related metagenomics projects. So what kind of information we can get from the analysis. And then for the metagenomics projects, what kind of post-project support that we can obtain. Let's say we are having the kind of the projects with Novogene or with NEXTGene here. So moving on, before we dive deep into looking into the data or the projects, so just some overview for whoever want to kind of rehash or refresh their minds on what is metagenomics. So basically the metagenomics refers to the study of microbial compositions of environmental or clinical samples. So for the samples that we obtain day to day or the environments that we have, actually there are a lot of microbes in the environment or in our guts. So this metagenomics basically is the study of these members inside the environmental samples or gut samples, and then to see who are they, and then ultimately we also want to know what are they doing inside the samples or inside the environments. So normally we try to kind of understand the microbial profilings. So who are they? And then through the metagenomics approach, this is different from the traditional culture approach. So we normally do not need to kind of do some isolation or do culturing in order to identify what are the members, what are the bacteria, or what are the microbes inside the samples. So this one for metagenomics, we are also able to identify some kind of useful markers. So these markers could be helping us to identify the virulence or AMR, the antimicrobial resistance, or some specific functions of those microbes inside the samples. And then potentially we can also identify maybe some novel species or novel microbes inside our samples, and then explore how these microbes kind of communicates or working in the samples, and then showing some phenotypes that we are seeing in the samples. So there are many ways to study the metagenomics or to study the microbes inside samples, a mix with different microbes. So most of the time we are looking at first what kind of samples that we can obtain. So two major types will be DNA samples and also RNA samples. So because the microbes are living organisms, contain the DNA and also use the RNA to express the genes or make the proteins that is the functional units. So when we are able to get the DNA or the RNA out from the samples, we are able to study who are they inside the samples and then what are they doing inside the samples. So based on the types of samples that we can obtain, different sequencing approaches can be done. So the very common way will be using the amplicon or targeted metagenomic sequencing. This is to kind of make use of some housekeeping genes or some conserved genes such as the 16S or ITS. So basically all the bacteria contain the 16S genes, and all the ITS will have the ITS regions inside their their genomics, inside their genomes. So we try to identify who are they based on these particular gene markers. So other than that, some scientists actually prefer maybe to use the shotgun metagenomic sequencing approach. So whatever we are able to extract from the samples, we try to sequence everything. So this is called the shotgun metagenomic sequencing. And there's another groups prefer to look on the RNA part. So they want to see what are the expressed genes or what are the functional genes that are inside the samples. And at the same time, also to identify who are they inside the samples. So this approach is called the metatranscriptomics approach. And actually, you will see the papers or the studies on the metatranscriptome will be slightly less compared to the other two applications. So this is not because it's less useful, but because most of the time when we obtain the environmental sample or gut-related samples, it's quite challenging to extract the total RNA that is of good quality and then proceed to do the metatranscriptome sequencing. So that is why you might observe the papers or the study on metatranscriptome is slightly less. Well, a lot of papers will be focusing on the amplicon or targeted metagenomic sequencing because this one, it has less kind of difficulties in terms of obtaining the sufficient samples to proceed for the sequencing. So it depends on what is our project goal or study goals. If let's say we are looking into the bacteria, and then most of the time we will be looking at the 16S amplicon metagenomic sequencing. And then if let's say our focus is more on the fungus, then we could make use of ITS marker genes. And then if let's say our project is of other goals or other targets like archaea or eukaryotes, there are also other marker genes that are possible to be used for the amplicon metagenomic sequencing. So for shotgun, most of the time we are looking into either short reads or long reads. So short reads, it could be using the Illumina platforms, and then long reads, we could be using the PacBio or the nanopore platforms. So actually, this is a very important step before we start any metagenomic projects. So to design a proper experiments, these are some of the suggestions in choosing the right applications for the projects. So most of the time, depending on the sample conditions, the sequencing specs that we are looking at, and how much data throughput that we want. So some of the design could be like, kind of the decisions can be made based on the flowchart like this. So maybe the first question we could ask is whether the samples is having high host contamination. So most of the time, because we extract the total DNA from a sample that is mixed with different types of organisms. So in some case, like the gut microbiomes, that we maybe extract the total DNA from the fecal samples or stool samples, then likely the samples will have high contamination of the host. So let's say it's human fecal samples, then we are expecting the total DNA extracted from the fecal samples will contain a lot of human DNA. While we are studying the bacteria inside the fecal samples, then the human DNA will be considered as contamination. So for samples of that kind of situations, normally we will recommend to go for amplicon sequencing. So amplicon sequencing, there are also two options. So to go for the full-length amplicon sequencing or to go for the targeted regions of the amplicon sequencing. So this is based on the platform that we choose. So, and then most of the time when we choose short read sequencing, because of the limitations of what we can cover on the amplicons, we are most likely not able to cover the whole, let's say, 16S genes, because the 16S gene is roughly 1.5 kb in size, while the sequencing technologies is only able to sequence, I think, 250 from one end and then 250 base pair from the other end. So overlapping some in between, maybe we are looking at around 400 base pair target regions. So depending on whether you want to achieve high resolution or you want to perhaps a more cost-effective approach, then we can choose either full-length amplicon sequencing or the short-read amplicon sequencing. So for the case where samples do not really have high host contamination, then we can choose between the amplicon sequencing and also the shotgun metagenomics sequencing. So of course, the amplicon sequencing is always easier in order to handle the samples and also in order to get the data out. And then the cost normally will also be lower. But if, let's say, we want to look into the genomes of the members inside the samples, then that will be the shotgun metagenomics sequencing. And then through the shotgun metagenomics sequencing, we will not only discover who are there inside the samples, we actually have all the genes of those members inside the samples. So we can know what kind of genes are contained in those microbes, and then likely what kind of functions are involved, and then whether they are showing certain phylogenetic relationship or sharing some ancestral kind of information from one sample to another sample. So more information that can be obtained from the shotgun metagenomics sequencing, but the cost normally will be slightly higher. So based on the types of sequencing that is used, of course, the analysis approach, there are quite a lot. So designing all these project actually is quite complicated, and then we need to do it before we start working on it. So if you want to have some discussion on how to design the projects, or based on the project goals, what is the best approach to go for it, feel free to reach out to us. We can also discuss further and then suggest or identify the best way on working on it. And so once we have decided how to go about it, then next thing we start working on it. So actually, when you have a proper experiment designs for the project, basically that is already half of the projects that is already half done. So the next is just hands-on. So what would happen during the hands-on for the metagenomics projects? So what you can expect will be, of course, first getting the right samples. So based on the selected samples, and then prepare or extract the total DNA. And then this one, after getting the samples done, then you can pass to NextGene, the person in charge. So we have the logistic team to help you to ship the samples to the sequencing lab. So in the sequencing lab, based on the sample manifest submitted, so you might need to inform the sequencing lab how many samples that you have, and then what are the labels that you have put on the tubes compared to what is the samples that you want to work on. And then the sequencing lab will not be immediately start on the sequencing part. So if they will do some QC check on the samples, and then to tells you whether the sample passed the QC, or maybe some of the samples is off risk, and then see whether you want to resend the samples or you want to continue with risk. So all this will be at the maybe the first 1 to 2 weeks time. So normally this step, because sometimes we are not so sure whether certain samples will pass the QC. So sometimes we will also encourage researchers to maybe prepare more samples and ship together in one go, so that if, let's say, some of the samples is not so good, then we have some backup samples that we do not really need to redo the sample shipments and everything. So it can it won't delay the projects per se. So once everything looks okay, Then the lab will proceed to do the library preparations, and then the library QC and prepare for the sequencing. At the same time, likely the sequencing lab will also request further information on the samples. So this one for those who have done a project like this, they might kind of receive a form which is called a BI form. So this is for researchers who subscribe the bioinformatics services. Then the sequencing lab would need to know additional information for the bioinformatics parts. So this is through the BI form. And let me try to maybe give you some examples of the BI form. So this is an example or demo BI form. So normally the sequencing lab will need some additional information like this one. Most of the time is halfly filled by the sequencing lab. So how many samples that they have received, and then they will also put in their IDs. So what is required from the researchers will be, how would you like to name the samples? So because sometimes when preparing the samples, the tube is so small, we are not able to write the full name of the samples on the tubes. So after this, maybe you want to give a proper names to those samples, then you can put the sample names over here. And then also to tell the sequencing lab what samples belongs to the same groups. So how many sample groups that you have, and then which samples and which sample are considered sample replicates. So we are having the list of samples, and then let's say we have A1, A2, A3, all belongs to the sample groups of sample group A. So we can put, let's say A1, A2, and then A3. So all these belongs to the sample groups of A. So with this information, then when they do the analysis, they will know which samples belongs to the same groups. And then when they do some analysis comparing different sample groups, then they will know what samples to compare to what samples. So addition to that, I think they will need to also some additional information like the choice of the database that we want to use. And also some comparisons. Let's say we want to compare certain samples against certain samples, or certain sample groups against certain sample groups. All this can be defined in the bioinformatics form. So this one is for projects that comes with the bioinformatics analysis. And then if let's say the project is on sequencing only, so no bioinformatics to be done, then likely you won't be seeing the bioinformatics form, or don't need to fill up the bioinformatics forms. So once the sequencing is done, then the data will be sent over for the researchers to kind of get, that will be the project deliverables. So if let's say the project comes with bioinformatics analysis, of course the analysis result will be delivered together. So this bottom here is just some additional information like what kind of DNA samples will be required for the metagenomic sequencing. So for the deliverables, what are the files that you'll be getting if let's say you are going for metagenomics sequencing projects? So for project without the bioinformatics service, the BI service, normally during the communication, you might also be seeing the project name with this particular tag is WOBI, so it's without BI. So for projects without bioinformatics and service, users will receive the samples or the PCR product QC reports. So this one to tell whether the samples is good enough to proceed for library preparation and sequencing. This one will be receiving, the user normally will receive it shortly after the samples are shipped over to the sequencing lab. So once the sequencing is done, then the users will also receive the data QC report together with the sequencing data. So both the raw and clean data will be provided. So no worries that kind of the formats of the data, because this one will be using the public or the common file format. So for Illumina sequencing, most likely will be the FASTQ files, and then maybe full-length sequencing like PacBio or ONT, you might be receiving the FASTQ file or the BAM file. And then for projects that comes with or this with BI service or the WBI, in addition to the sample QC report and data QC report, so the full analysis report will also be provided as the project report. So in addition to that, sequencing data, raw and clean, will be provided. together with the analysis results files and data. So I think somehow we received some queries on whether we will be providing the sequencing data after the project. Yes, we will be providing the sequencing data, raw data and clean data for all the projects. So no worries that we will keep your data as hostage. So once you have the metagenomics sequencing projects, you will be receiving the sequencing data raw and clean. And yeah, so how this data will be sent over to you. So for Novogene, they have a delivery system, data delivery system. So you will be receiving a email with batch ID and also password. So and then the link to their data delivery system. So you just need to click the link and then put in or kind of log in using the batch ID and password. And then once you are inside the system, then it's like normal cloud platform like Google Drive or the OneDrive. We just need to click and download whatever files that is shipped for the project. So once the data is downloaded in your computer, most of the time you'll be seeing multiple files like this. So for Novogene, they will be trying to reduce the number of files delivered in a project, because if let's say metagenomics project, there might be easily hundreds to thousands of files. So to make the user easier, they will zip all the results into a zip file, so that when you download, you just need to download one single zip file. But the caveat is that sometimes when the project's involving a lot of samples, then the file size could be quite big, so it might take some time to do the download. So once we download the files like this, let me show you how it looks like. Yeah, hopefully my file explorer is big enough for to be viewed in this Google Meet session. So normally the download will include the main file, so the zip file. So normally the files will be the biggest among all. So there are some miscellaneous files that can be downloaded together in the session. So these are basically some additional information in case you really want to check whether your file download is completed without any problems. So they provide the MD5 files. So this is just a very short, very simple text file that looks like this. So this is the MD5 values for this particular results files. So we can use the programs provided in order to check the MD5 for this particular file and see whether it matches the MD5 files provided. So if let's say this file is not downloaded fully, then the MD5 for this file will likely not matching the one provided inside this particular file. So in that case, you can try to redo the download. But for me, because the download file is a zip file, so most of the time, if you are able to unzip this particular file, then the download is considered complete. So unzipping this file in Linux or Mac computer, they do come with the built-in functions for that. So you just need to use the unzip commands to do the uncompression of the files. For Windows, now we do have the built-in functions for that, so we can even directly click into it to view the files inside. So this is a good functions that is provided by Windows, but somehow because the files was zipped using Linux functions or some Unix functions, so even though Windows have no problem to open it up and take a look at files inside, but if you want to unzip using the built-in function like extract all, then likely it will end up with some problems. So the recommended methods will be using software such as the WinRAR or the 7-zip. So if you want to unzip this particular file, and then this is the two programs that we suggest to use in Windows to do the uncompressions. So once the uncompression is done, then we will have all the files inside. So here, just let me try also to show you what kind of files that we are seeing. So for one project with a BI service, we will be receiving a folder that contain a lot of files and data. So for users who first look into this folder is going to be quite overwhelmed. because we don't know where to start. So most of the time, I will suggest to start from the report. So every project comes with a report. So inside the report, there should be one HTML report. So double click on this, supposedly to open it in any web browsers. So, because my files here is still in the compressed form, so if I open it in the web browser, likely it's going to be not complete. So I won't be showing you the one here. So if I want to try and compress everything in Linux or in Mac using the unzip command such as this one, results. Yeah. So we just need the unzip command and then followed by the file name, then and enter, then we will be unzipping this particular file. So before that, we need to make sure we are in the right folder containing the files. So if you're using Linux or Mac computer, you just need to open up the terminal and then navigate to where the files is located and then use the unzip command to do the uncompressions. So once executed this particular command, it will try to unzip whatever files inside. And because metagenomics do contain a lot of results files, so this process might take a while. So I'm going to minimize this particular one. Go back here. So we supposedly to see all the files uncompressed once it's done. Okay, so yeah, this is what we can expect when we have metagenomics projects and all the results file delivered to us. And next, I will be showing you the analysis reports. So let me open up the analysis report. Okay, so this is how the reports looks like if we open it up in a web browser such as Google Chrome or maybe Safari or Firefox, whichever web browsers that you prefer. So it's a HTML format. So these reports normally it starts with the overall project information, like what is this report about, and then what is the primers that is used for this amplicon 16S sequencing. And then this particular report is actually working on the 16S V4 regions. So some of the project information can be obtained from the very beginning of the reports. So subsequently, in this report, we do have some informations on the library constructions and sequencing that is done in the lab. So this could help you when writing the manuscript. And also it will have the overview of what are the analysis that has been conducted for this project. And then importantly, there will be analysis results and then appendix and references. So it's going to be a long report. So let me try to scroll slightly, kind of scroll down to look into the reports. So introductions, what is metagenomics or 16S amplicon sequencing and etc. So the project workflows starting from the sample preparation, do the sample QC before the library construction, library QC before the sequencing, and then do the data QC before the data is submitted for analysis and delivered to you. And then next will be some general or some quick overview on the library construction and sequencing, how it was done. And next, importantly for bioinformatics analysis, so this is the overview of the whole pipelines or whole methods that has been used for this project. So of course, they feel free to go through the write-up over here, but most of the time I prefer to look at the workflows over here. So starting from the sequencing data, the bioinformatics have been carried out to do the data QC, so merging the reads and then filter the text and then do some chimeras removal. So this is the part of the QC step to ensure we have good data before we're looking into the metagenomic text. So once the QC is done and then we have good quality ASV clusters or ASV text. So this is our some people prefer to use the words of OTU, operational taxonomic units, and then now is alternative, sorry, it's called the sequence variance. I forgot what is the A representing. And compared to the previous OTUs, because OTUs most of the time it's used to refer to the sequence that is whenever we are seeing the sequence having 97% identity, we are putting them together as same sequence cluster or same taxonomy unit. So, and then we are assuming every taxonomy unit represents one species. But in situations like whenever 97% identity, we are consider them as a single species, that is not so logical. So that's why we have ASV most of the time now, because this ASV will try to exclude the sequence that likely come from the sequencing error, and then whatever that is remaining, then we try to see how many different sequence cluster or sequence variants are there. So we no longer use the 97% cutoff to group the samples. So after having the ASV, then the subsequent analysis will be carried out based on the ASVs. So this include the typical workflows on taxonomic classification, so to see what are the species that is inside our samples. And then this involve also the phylogenetic tree to see the overall kind of the members inside, and then how those members are related one to another. And then of course metagenomics, we want to look at the samples alpha diversity and beta diversity. So alpha diversity is the I would say intra samples variations or intra sample diversity. So in one sample, we could have a kind of small number of members. We can also have many different numbers of members. So when we have one sample that contain a very rich community, so it contains many different types of species, many different types of bacteria, then we consider it of high diversity. And then for samples that may be containing only a few different species, then we consider those samples as low diversity. So this is alpha diversity analysis. And if we want to compare across different samples or compare different sample groups, then we are comparing the diversity in terms of beta diversity. So this is the cross sample or intergroup variation analysis. And then we are using different approaches or different methods to calculate the alpha diversity and beta diversity of the samples. And then further on that, we can also compare statistically what are the species that are different across different sample or across different sample groups. And then we can also try to identify the species, the groups to groups differences, how different is sample A compared to sample B, and then how different are sample groups A compared to sample groups B. So we will have a statistical numbers or metrics on those comparisons. So other than that, of course, we can do more from alpha diversity and beta diversity and comparisons. So for some projects, when we want to look deeper inside, we can also perform some functional predictions. So most of the time for 16S or the amplicon sequencing, we know who are they, but what are they doing is based on the predictions. So these predictions, it can be done based on several tools listed here. And then other than that, we can also try to check the microbial compositions with their environmental data. So how is that environmental factors affecting the microbial distribution inside the samples? And also we can perform some additional analysis such as the network analysis to see how the species are kind of connected or correlated to one another. So whether there are certain species always co-occurs or always present when another species is there, or maybe when this species is present and then this species will not be inside the samples, and etc. So you can see the colors in the flow chart here in green color. So most of the time for projects, standard projects, without any further request, the project will cover all the way from data QC to taxonomic classification, alpha diversity, beta diversity, and comparison across sample or sample groups. So for more advanced analysis required, then we can also include that inside the project. So this is just an overview. So the analysis results parts basically will summarize all the analysis that have been carried out for the projects. So for the full data, actually we can also see where they put the raw data. So most of the time they will tell you for this particular part, sequencing data processing, where they keep the data. So it's inside the result folder, and then folder 01, and then according to the samples, and then the data is arranged or ordered in such a way. So that's why these reports also act like the maps for us to navigate across the whole result folder, which is containing a lot of files and very big in size. Okay, so let's go through the analysis briefly one by one. So for analysis, since the first step will be the data QC, so basically the 4.1 is on the data QC summary. So how much data that we have obtained for each of the samples. So for this particular report, we have 76 samples involved. So these samples starting from A1 all the way to A15, and then if we switch to the next page, we are seeing A16 to A30, and then to all the way to A36, and then we have B1 all the way to B20, and then C1 all the way to C20. Yeah, so 76 samples, and then I think likely there are 3 groups, so A, B, and C. And then we have 36 samples for group A, and then 20 samples for group B, and 20 samples for group C. So for each of these samples, inside the table, we can see how much data have been generated in the sequencing. And then this will be depending on how much data that we subscribe for the projects. So most of the time, it depends on the sample types. So for example, like gut microbiome samples, we are expecting to have a lower diversity, not that much of bacteria inside. So maybe we can get maybe around maybe 30 to 50,000 of sequencing tags to start with. And then for samples from the environments, for example, like the soil sample, most of the time it contains a lot of bacteria inside. And then if you want to really have a very good picture of the samples, then likely we will go for higher sequencing throughput, perhaps more than 100K tags per samples. So in this project, we are actually seeing every sample is having more than 100,000 PE tags. So it's relatively good coverage or good sequencing coverage for each samples. So from this raw sequencing data, how much after the pipelines pre-process it into the good quality one. So it try to first combine. So we have forward and reverse reads, and then we try to overlap it, combine into the text. So one text represents one kind of the sequencing sequence for the samples. So how much is considered good quality and then qualified. And then because we are using primers to amplify the particular regions or particular genes, so this one will check whether the sequence after overlapping, they are having the primer sequence at the front and the end. So whether it's a full target or full amplicons from the samples. And then it will also try to check whether there's any chimeras. So chimeras is kind of maybe it's mix of one samples and with another samples in the same sequence. So this is considered a chimera, then it will try to exclude it. So the remaining one, so this is the non-chimera one. So we have around this number of tags remaining, and then this total data throughput. So for some samples, you might see slightly lower number of qualified or the high quality tags or clean tags over here. So this normally will be linked to the sample quantity and also the PCR cycles that is used to amplify the amplicons. So if let's say the sample is of lower quality, lower amount, then the PCR will go for more PCR rounds, PCR cycles. Then likely it will generate more and more chimeras. So after the filtering, then we will see less number of non-chimera tags at the end. So if let's say we really want to have good quality data on our samples, then the recommendation is always having good quality samples and then good kind of sufficient amount of samples so that we can reduce the PCR cycles when generating the amplicons to be put inside the sequencing machine. So, and then from the average length, we can also check whether the length is of our expectation. So this one is V4 regions. So after minus off the primers front and end, then this is actually the expected amplicon size. So the V4 regions is roughly this particular size. And then GC percent for 16S amplicons normally is around 50+%. So it looks everything is acceptable or within the acceptable range. And then Q20, Q30, this one are the quality score. So normally we are expecting the quality score Q20 more than 90% or 95% based on our current technologies. And then Q30 normally raw data normally we're expecting to be more than 80%, but for the technologies right now and then how the labs handle the sequencing, I think most of the time we can achieve more than 90% or even more than 95%. So if we are seeing some of the sample is having low quality, then likely some problems might could happen during the library preparations and things. So most of the time, sometimes we might need to go back to the sequencing lab to check whether certain samples is having some problems during the library prep. So if let's say we are seeing everything okay, so their number of reads, number of tags, and then their rate, the text length, amplicon length, and GC percent, everything looks normal, then basically the data is sufficient or good enough to go for the next step. So next step is the OTU or ASV analysis. And this part, the results is actually the kind of, in this report, it will be put in into different visualizations methods such as the heat map, the bar plots, and everything. And the full results is actually inside the folder 02. So if you want to have the full sequence, kind of the total ASV and their sequence together with the taxonomic classifications, normally we need to go to the result folder to obtain on it. So it will have multiple tables on these particular results, and then to quickly help us have a kind of overview of the samples content or who are they inside the samples. Normally in the reports we will be seeing different visualizations methods, so like the bar plot, so we can see all the samples is having a highest percentage on the Firmicutes, and then some samples also contain relatively high I think is it Bacillota, yeah. So in the reports, when we have a lot of samples, the pictures might be a little bit running out of their original shape. So if you want to have the full high resolution pictures, normally each of these pictures that we are seeing in the report, the full pictures or the high resolutions one will be also provided in the project or the results folder. And over here is just a quick overview of the sample contents or the members microbial community inside the samples. So this one could help us perhaps to quickly identify whether any of the sample looks like outliers or very different from other sample replicates. So from there, we can also see whether certain samples is very different in terms of their phylums contain, class, or orders, or different based on the different taxonomic rank. So you can see on the reports over here, for metagenomics, they will provide the results for each of the taxonomic rank. And then the name actually is shortened to P, C, O, F, G, and S, which represent the phylum, the class, the order, the family, the genus, and the species. So all these pictures, all these results, whether it's tables, everything is inside the results folder. And then in this report is just showing you a quick summary on all the analysis results. So next, if one kind of interested to know how many of these ASV is shared across different sample groups, how many is discovered across all the sample groups, the results can be, we can have a very quick overview over here. So unique to group A, and then unique to group B, and then share across all the group A, B, C. And then if let's say we want to know exactly what is this 1011, normally we need to go back to the results folder, look for the tables for this Venn diagram, and then look for the one that shared across all the samples, sample groups. And then how we know kind of what to put in the Venn diagram and also perhaps the flower diagram like this. It's all based on the BI form that initially submitted to the lab. So if you want to compare group A versus group B versus group C in the Venn diagram, we can, I think over here, yeah. So the Venn diagram, we want to compare maybe A versus B versus C. And then we also maybe want to compare perhaps A1, A2, A3, A4, and also other types of different combinations. We can define what we want to put in the Venn diagrams by specifying them into the BI form. So all the analysis in the report is actually based on the specifications that we put into the BI form. So yeah, so this 4.1 and 4.2, basically the data QC and also a quick taxonomy classification overview. And then this one is based on the taxonomy phylogenetic tree. So we can see which clades or which groups is highly expressed or highly having the high abundance inside different samples. So this one, yeah, again, is a quick overview. So full results will be inside the results folder. And then the ternary plot. So this one is specifically useful for experiments that involve three conditions. So ABC, for example, like in this case, and then the size of the dots represents the abundance across different samples. If let's say you see the dots is closer to A, that means this particular taxonomic groups is actually found more to be inside the samples group A. And then if let's say it's closer to C, then it is found. And then if let's say the bigger the size is found a lot, and then is present in a lot in group C. But if let's say it's in the middle, that means it's found basically quite average across all different samples, different sample groups. Yeah, so quick overview of the samples. Next, we will be looking into the alpha diversity. So this one, we want to see whether certain samples is having a high diversity or low diversity. So there are different ways to measure the alpha diversity for each samples. So including like the one that you are seeing here, the child one, the dominance, the good coverage, observed features, Shannon, Simpson, and etc. So all these are different methods in measuring the alpha diversity. So it depends on what we want to look at. So most of the time, I think the most straightforward one will be the observed features. So this one tells how many different species that is found inside the sample. So for example, A1 found a total of more than 1,000 different species inside, or ASV inside the samples. And then A2 and the rest is actually seems to have less. So this one can directly tell us whether the samples is contain a lot of different species, or actually is actually only having a low number of species. So, and then other than that, I think the more commonly used would be the Shannon and also the Simpson. So when the samples is having a lot of different species, are those species present like evenly? So let's say we have 10 different species inside the samples. Are we seeing roughly maybe 10 to 20 different percent for each of the species? Or are we seeing 90% belongs to one species? Well, the rest of the species roughly contribute to maybe a few percent or 1 to 2% of the whole samples. So this is we consider the evenness of the samples. So the Shannon and Simpsons normally will also measure the evenness. So if we see the Shannon and Simpson to be of high values, normally that means the samples or the species is distributed quite evenly across different species. And then if let's say we are seeing a very strong dominant species inside the samples, and then normally the evenness will be lower. So this is some of the important metrics that we normally will be seeing inside the alpha diversity analysis. So other than that, actually another important things to look at in the alpha diversity will be this rarefaction curves. So this one actually is also look at the diversity, and then it plot for each of the samples. So one line represents one samples, and then it tried to see how many species can be discovered when we subset the samples. So based on maybe 10,000 tags, how many species we can discover? if we increase the number of tags to maybe 20,000, how many different species that we can discover in the samples. So this, after we plot the information out such as this way, then we can tell whether the sequencing is saturated or is having good coverage to reveal all the species inside the samples. So from here, you can see all the samples is kind of become the flat lines. So that means even though we increase the sequencing more and more, it won't help us to discover more species inside the samples. Basically, the sequencing have achieved the saturations. So the sequencing is having good coverage to cover all the species inside each of these samples. So of course, we can also view this rare fraction curve based on different alpha diversity metrics. So most of the time, as long as we are seeing is approaching flat lines, that means likely we are not able to discover new species even though we increase the sequencing coverage, getting more data. But if let's say it's still increasing, is at the log phase, then that means if we keep on sequencing, likely we are going to discover more and more different species inside the samples. Okay, the next one is the species accumulation box plot. So this one, just now we are comparing different sequencing throughput. So whether we keep on sequencing, whether we are discovering more and more. This one, the x-axis is the number of samples. So in total, we have 76 samples. So how many species we are discovering when we are having only one samples? If we are having 10 samples, how many species we can discover across all the samples? If you are having maybe 50 samples, how many species we can discover? So this one will tell us whether we have sufficient sample replicates or samples to review all the species in the total samples environments. So if we are seeing these plots is not flattened, then that means if we keep on increasing the number of samples, likely we are going to discover more and more species across all the samples. Yeah, so and then other than that, this is the rank abundance curves. So this one can roughly tell us whether the samples contain major dominant species or they are having kind of evenly distributed species. So if we are seeing, supposedly if let's say we have 10 species inside a sample and then they are having the equal abundance, then we should see a flat line over here. If we see kind of one very dominant species, well, the rest is only rare, kind of low abundance or very rare species, then we should see the lines drop very quickly vertically over here and then move sideways at the kind of bottom of the graph. So this one will tell us if let's say it drops very fast, that means there are one or a few dominant species, while the rest is actually distributed, kind of having low abundance across the samples. So if you are seeing kind of like this, so it's considered there are some species having high abundance, while the rest is having low abundance. Is like, I would say, normal distribution. So this one, this just want to have a quick look whether certain samples is having very strong dominant species inside the samples compared to other members inside the samples. Yeah. So this alpha diversity, those are the information that we can get from the alpha diversity. And next, beta diversity. So like mentioned, so alpha diversity is comparing within the sample. So we want to see sample by samples, how's their diversity, and then whether they are evenly distributed and whether they are certain very dominant species inside the samples. So next, I think most of the projects involving multiple samples, we want to see the differences or the changes across the samples. So for example, like this particular report is having 3 groups. So most of the time when we are having 3 groups, we want to see what are the difference among the 3 groups. So this alpha diversity differences, first is this, because when we are measuring the differences, we are looking at their difference. So weighted and unweighted, it depends on, because we have different species, and then different species have different abundance values. And when we compare, we are comparing different species across based on their abundance values. So, and then measuring the distance for metagenomics, normally we are seeing weighted and unweighted. So what is weighted and unweighted? A quick explanation for the weighted and unweighted is that weighted will consider their abundance inside the samples. Well, unweighted, basically they are comparing whether the species are present or absent inside the samples. So when we are comparing for species one, whether they are absent or present in sample A, and then whether they are absent or present in sample B. So it's like kind of a binary comparison. And weighted, it will compare species A and then inside sample A, whether they are having high abundance compared to sample B, whether they are absent, present, and having high or low abundance. So the comparisons, when they are considering the abundance values, then it's considered the weighted. So this one, it include both weighted and unweighted. So the based on the explanation over here, the heat map based on weighted. So the upper part is the weighted unifact, and then the bottom parts, the yellow part, is the unweighted unifact distance. So sample group A compared to sample group C for the first square over here. So based on the weighted difference, so A and C is roughly 0.13 difference. And then based on absent and present, so we can see quite a lot of species could be absent or present in one sample, but not in other groups of samples. So this one, the higher number represents the higher distance. So you can see when we compare A versus A, is going to be 0 because there's no dissimilarity or no distance across the samples supposedly. So other than that, we can also compare according, kind of put it into the cladogram such as this one. So group A, group B, and group C, what are their major bacterial groups, and then how are their abundance overall across different sample groups. So from here we can see actually the species or the organisms or microbes discovered inside group A, group B, and group C, their compositions are more or less the same. It's just that having I think group A is having more bacteriota, and then B and C is actually sharing quite similar patterns over here. The biggest group being the firmicutes, and then followed by the bacteriota and etc. So this is this gives a quick overview comparing different sample groups. And then most of the time, we will be also looking into the PCOA to tell or to see the distribution across all the samples, putting them into one putting them into one space. So the red one is group A, and then the blue is group B, and the green one is group C. So you can see actually they are not that much difference comparing different sample groups. It's just that I think sample group A is quite close within each other, and then the rest B and C quite overlapping with one another, and then the distance within the sample is also larger compared to the group A. So this one, from this particular PCOA plot, I would say all the samples are sharing similar microbes groups inside the samples. It's just that their abundance value could be differ slightly. But overall, from here, I don't really see very much difference between group A, group B, and group C. So for the cases where group A, group B, and group C are very different from one another, inside the PCOA groups, PCOA plots, we will be seeing group A samples all located together very close to one another, and then the group B will be forming another cluster, and then group C will be forming another cluster. So if you see this kind of patterns separately for each of the sample groups, then this will shows that each of the samples group is very different from another. So, and then for this one, when we are seeing all the samples are overlapping with one another, this will show that actually they are quite similar across their microbial profiles. So for the same plots, we can also visualize it in the 3D form. So this one with the files provided is going to be interactive. So you can change the color, you can view the plots in 3D form and etc. So interactive. But inside this report is going to be showing you the screenshot of the picture. And next, PCA. So PCA is useful for the same purpose when we want to compare across different samples, but when we are just looking kind of a quick overview of their distance, so we do not really consider the taxonomic informations, because for the sample, supposedly, let's say we have 10 species, but all 10 of them are bacillus, and then another samples, maybe all 10 of them is a mix of lactobacillus and also brucella and etc. So PCA will only consider whether they are sharing certain same species. If let's say bacillus and lactobacillus, even though to us they are supposedly to be close, but for PCA they don't consider it to be close. It's because bacillus is different from lactobacillus, so that's why they are considered it to be different. So when they are considering the differences in different way, then the plots will be different. So for metagenomics, I think the more commonly used method will be the PCOA and as well as the NMDS. PCA, I would say, is just a side reference. I won't be looking very deep into the PCA plot for metagenomics. For maybe other applications, this PCA plot will be useful. For metagenomics, I think it's not that suitable. So for NMDS, this is also the similar approach compared to the PCOA. We'll consider the relationship across different taxonomic unit. And yeah, this one again, we can see whether certain sample groups are forming clusters and then they are different from other sample groups. So this one again, it shows that the samples dots are all distributed across the whole plots, and then there is no distinct separations of different dots. So it seems to me overall, based on their microbial profiles, they are actually not that much different. So this is a quick overview when we put all the samples into the plots. So how exactly different from one sample groups to another sample groups, then we need to run some statistical models or do some calculation to tell us how different are different or how similar are similar. So this will come to the next sessions. So the community differences. So the adenosine, the MRP, the adonis. So this will tell us from comparing sample group A and group B, how different or how close they are based on the R value and P values. So P values, of course, the lowest, that means the difference are significant. And then R values will be how close or how different comparing different sample groups. So here, quick summary, group A and group B seems that they are significant differences. And then group A and group C also seeing differences across these two groups. And B and C, the p-value is quite high. So based on the calculation, B and C have totally no significant difference. So if we cross-check these results with the plots again, so group A and then group B and C. So group B and C, the distribution is kind of across whole plots. And then group A somehow is close to one another. So intra-groups differences is low compared to group B and C. So yeah. Normally this part, the results is to be kind of interpreted together with the beta diversity analysis. And then there are different ways of measuring the differences. So this one is based on the analysis of similarity, or the program is called the NOSIM. And then if we look at the different statistical model in doing the measuring, then we are also seeing some slightly different numbers. So it depends on which one is more bringing out the story for your paper, perhaps. Then in the paper, normally we can choose to report one or two, and seldom we will be reporting everything. So the next one, Adonis. So this one is actually using the methods called the permutational MANOVA. So the software is called the Adonis. So some people will call it the Adonis analysis, but supposedly it's actually this particular statistical models that is used to do the comparisons. So again, it used to calculate how difference from group A to group B, group A to group C, and then B to C. So yeah, from here we can also roughly tell how different are they. So this one, A and B having higher differences compared to the B and C. And next, if we want to look at which species is contributing to the differences comparing different groups, then the subsequent analysis can help us identify the species that are different comparing different sample groups. So the first one is the simple. So this one will highlight, kind of show you how's the distribution or the abundance of the particular class of bacteria inside each of the samples. So this one particular, I think the bacteroides is the one that contribute most variance into the samples or group differences over here. So again, this is a quick summary on look at the differences. And of course, we can compare particularly different sample groups to tell which sample groups is having totally different abundance values, comparing, let's say, in this case, C and A. So bacteroides, we can see actually present a lot in the sample groups A. And then the group C is having low numbers of these bacteroides. So the confidence intervals or the p-values will show how significant is this calculation. So t-test, similarly, we have I think this metagenome seq. So this one is also doing the pairwise comparisons. So it's just different methods to do the comparison. And I think this is the most widely used approach to identify the species or the organisms that is kind of stand out in the comparison. So which particular is difference across different comparison and across different multiple groups of samples. So if let's say we want to see which one is contributing to the ABC differences, and then we can look at this one. So in some case, like the B and C, just now we have seen actually B and C is quite similar. So when the software do not discover any species that is differentiating B and C, then it will show no results like this. Yeah, so this is again the different representations of the left SE results over here. So just now we are seeing the plots by the different bacterial groups inside here, and then how different are they or how significant are they. And then this one is trying to put it into another visualizations to see how's the overall groups and the subgroups inside the samples. So this summarize on the alpha diversity and beta diversity. So tells the differences within a sample and tell the differences between samples or between sample groups. So the next one we want to see, when we see differences, what are the functions that are involved? So biologically, how different are they inside the samples? So this one, if I continue to scroll and scroll and scroll, this all will be belongs to the functional prediction. So this is a very long section. So instead of showing you one by one, let me summarize these particular sections in one slide. So functional and phenotypic prediction tools, they are actually the whole sections. I think more than 20 plus different subsections there reporting the results on these functional or phenotypic predictions. So summarize everything, they have used multiple tools to predict the phenotypes or the functions involved in the samples. So basically will be the PyCrust or PyCrust and basically and PyCrust 2. So they use two versions of the PyCrust to do the prediction. And then they also use another program called the Text for Fun, and then the bug-based and FarProtect. And then if let's say this is a report for fungal ITS reports, then we'll be using the FunGuild. So basically they are having the same functions, just that PyCrust and PyCrust 2 is suitable for all kind of samples because they compare the species inside and then what are the pathways, biological pathway that can be carried out by those species. And then Text for Fun is more curated towards gut and soil samples. So if let's say the samples is from water or from other habitats, then Text for Fun results might not be very suitable or might not be that comprehensive. So then bug-based specifically is predicting the functions for gut samples. So more or less kind of the functions predicted is related to gut pathways or gut biological processes. And then the FAR-Protect is for environmental samples, is like the nitrogen fixations, and then the whatever that we normally, the pathways that is involved for the environmental, like dealing with the heavy metals, ions, and etc. So based on projects, you can be more focused on certain groups of the taxonomy or certain groups of the functional predictions over here. So for each of these analysis, because when they run the analysis, they just run everything for you. So it's up to the researchers to pick the right results to be further analyzed, further interpreted, or further reported in the papers. So for each of these programs, inside the reports, actually they have included the relative abundance bar plots and display the this one display the top functional pathways per sample, and then display the heat maps. What are the pathways across different samples, and then the PCOA, the Venn diagram, and also different plots to show which functions is more represented in certain samples. And then these functions is based on different tools. So yeah, depends on the projects. You might be picking the right tools or right results to go for it. If you go to look at the reports, scroll down, I think it will also need some time to scroll all the way to view all the results for the functional and also phenotype predictions. So yeah, so pick up the right tools or right data sets, and then the rest, all the results are provided in the results folders. So the rest, if let's say you're having gut samples, then you can totally forget whatever inside this particular FAR-Protect annotation. Okay, so last two parts are actually quite optional. So if you are interested to look at which species is more correlated with another species, whether certain species always comes together, or maybe certain species always missing when another species is there, then this can be observed through the correlation analysis. So particularly it provides the results in network, 2D and also 3D. So these 2D plots, it will show you which particular groups of microbes is having kind of a high correlation with other species. So when they happen, when they are there, and then most likely the other types of species will be there, or maybe some other types will be missing from there, and etc. So this is the 2D form, and then optionally, if you're interested in looking in the 3D space, they also provide you with the interactive HTML files to view the correlation across the genus or species, different taxonomy rank in terms of the different kind of in the 3D space. So this is on the correlations. It don't really relate to the sample group. So from here, normally I won't be able to tell whether this correlation is related to sample A or sample B. It's just which species is related to which species. Okay, lastly, the environmental association analysis. So even though this is considered, the name is environmental, but actually it's an associations analysis where because our samples, this one is quite straightforward. We have 3 sample groups. We are comparing sample A, B, and C. But in real situation, perhaps we are gathering samples from a field, or gathering samples from hospitals. We are gathering samples from a school. And then we have additional information, like let's say we gather the samples from a school or the students. We have their BMI, we have their age, we have their additional information. And then we want to see whether any of these variables is correlated to the microbial combinations. So in this case, we can put in the additional information to be analyzed together with the microbial compositions. So additional analysis can be carried out if we have the additional metadata of the same projects. So this one, for the same samples, to do the comparison, we need to have both set of data for the same samples. So let's say for this project, we have 76 samples. So we have gathered the microbial compositions for the 76 samples, and then we need to have the information for the 76 samples, same sample sets, same, so that we can do the comparison or the association across it. So due to the time constraint, so I won't go very deep into this. So if you have some experiments that you foresee this could help, then feel free to also subscribe this particular analysis. This one, you will need to supply the metadata for the bioinformatics team to work on the association analysis in this part. And then end of the reports, it shows basically what are the steps and programs that is used together with their parameters. And then details methods write-up, especially useful for publication purpose, is inside another PDF files. And then how to deal with different zip files provided, and the reference that is used to conduct the analysis. Yeah. So before I end the session, I just also want to show you that for the metagenomics projects, basically NextGen provide the tech support, I think up to 3 months. So in case you need some further helps after you get your data, we are here to help you download the data, to help you to go through the results together, and to help to identify which files that likely contain the data that you want, and etc. So other than that, because experiments are difficult or complex, sometimes after we see the result, we may observe maybe some of the samples doesn't seem to match so much inside the projects anymore. We want to exclude them, or we want to rename some samples and redo the analysis, and etc. For Novogene, it do provide platforms for users to redo the analysis or to customize the analysis. So this is called the Novogene. So for the projects that is conducted in Novogene for amplicon metagenomics and also RNA-seq, users can make use of this platform. So this include like, let's say we want to rearrange the samples, rename the samples, regroup the samples, and etc. We can manage the sample information over here. After we kind of reclassify or reannotate the samples that we want, and then we can redo the analysis listed in the report by ourselves. So we can fully customize whatever that we want to run over here. So let's say I want to redo the left SE analysis, and then we can regroup the samples, and then what are the samples that we want to include in the analysis. I think we can also adjust the image, like how big we want or what is the font size that we want. So it's fully customized. And then when everything is run, so we can do the analysis over here. And then we can also customize our own analysis reports after we get everything done according to what we want. And it also provides some tools for users to do some customized plots. And also, I think they have incorporated an AI agent over here. So for different agents. So if, let's say, you want to plot certain things, but you are not so sure how to plot it, maybe it's a combination of different plots. So it's not directly available inside the toolkit, then you can maybe upload the plots, example plots to this bioplot, and then ask the AI agent to help you prepare the code in order to plot the data that you have. So this NEXTalk is similar to like ChatGPT or other AI chatbot. And then, this one is the one that if you put in an image, then it will try to help you identify how to redo the plots for your data. So this one is the R-based. So if you ask the agent to help you do certain plots, you just need to provide the data for the agent, and then tell the agents to do a bar plot or do a Venn diagram or do anything listed over here. So, and then it also have an agent to help to do the paper polish. So feel free to try it if, let's say, you have certain projects that are working with Novogene or NextGen over here. Yeah, so at the moment, I think there's no additional charge for these particular platforms if shall you need to redo certain analysis or customize your own analysis. All right. So summarize everything. So we have gone through how a project can be designed, and then what we should expect during the project, and then what we should expect at the end of the project. So we have also gone through very comprehensively on the analysis and their results, how they looks like, and then after the project, what you will be getting in terms of the technical support. So thank you so much for staying with me all this, I think almost 1.5 hours. Yeah.
Okay, thank you, Jiaxin, for the comprehensive bioinformatics analysis report walkthrough. So now we will open the floor for the audience for the question. Does anyone have any question for Jiaxin today? Feel free to unmute yourself and ask, or you can type your question in the chat box. It seems to not have any questions from our audience. Yeah, so before we wrap up, we would love to hear your thoughts for today's session. If you have a quick moment, please fill out the survey form in the link in the chat. Your feedback is valuable to us, and we will also share the e-certificate with you once it's ready. Also, if you have any extra question after this session, please feel free to contact us at sales@nextgene.my. So once again, thank you again, Jashi, and all of you for joining us today. We really appreciate for your time, and we look forward to connect with you guys again in the future events. Thank you, everyone. Have a great day. Bye-bye.